In Chapter 6, we're going to talk a little bit about gene interactions. So unfortunately, not everything is as simple as those first two chapters we looked at with Mendelian ratios, monohybrid, and dihybrid crosses. A lot of times, it's a web or big complex network of genes that interact together to influence phenotype. So we'll start to look at some of these more simple cases of gene interactions here in Chapter 6. Again, here is the chapter outline for your reference. So one way to see if something is a complex trait, that is a trait that is influenced by multiple genes, is to kind of look at the phenotypes produced. So when you have something, for example, colors of peppers, you have a whole range of different colors for peppers. Not only can you have green and yellow and red, but you can have shades of green and yellow and red. And so generally by looking at the number of phenotypes of this trait, or of different options for this trait, so in this case, colors of peppers, the more different colors there are, the more genes that are behind that, right? And so we kind of saw that earlier, I think it was in chapter 3, when we looked at coloration and having dosage of alleles. So if you had more copies of the gene that led to that phenotype, it kind of spread out the bell curve more, and the more spread out the bell curve is the wider range of colors. And so instead of just being a range from white to red, or whatever our example was before, you have a whole bunch of different colors here with these peppers, and that means a whole lot of genes are underlying this trait. So the first layer we're going to add on top of this Mendelian inheritance that we talked about in the first couple chapters is haploinsufficiency. So in a normal homozygous individual, homozygous for the wild type gene, we have two copies of the gene, one from the mother and one from the father. So this is assuming a diploid individual like a human or a dog or a lot of the mammals. So in this individual who is homozygous for the two dominant genes, we end up with two functional proteins, one from parent 1 and one from parent 2. Now in heterozygous individuals, we have two copies of the gene once again, and we have one copy that is just like that homozygous dominant or homozygous individual that had the dominant allele, where we get this functional copy of the protein. However, we also get a second copy of this gene that is producing a non-functional copy of that protein. So in this heterozygote, the dominant allele or the dominant trait is the phenotype that we see. So it's enough, it's haplosufficient. So having half of the genes be functional is enough to display that phenotype in this individual. In an individual that is homozygous recessive, we have two copies of this non-functional gene that are creating non-functional proteins. And in that individual, we don't see the dominant phenotype. That's where we see that recessive phenotype. So we're going to look at a few different examples of haploinsufficiency. So at first, I want you to focus on this first column. Ignore everything to the right at first. So in this plus individual, this is our wild type genotype. So we get two of these globular proteins here that are absolutely normal, and the resulting phenotype that we exhibit in the individual is wild type. Now, in the second individual, this is the mutation. The M's are the mutation in this case. We have two big M's and 0 of those proteins, the wild type ones we saw on the row before, are created. And thus, the phenotype of this individual is going to be mutant because he has two of the mutant alleles. Now, generally, if we have a heterozygote, like we talked about with the first two chapters that we did this quarter, usually having one copy of the allele is enough to show that wild type phenotype. However, if this gene shows haploinsufficiency, then what happens is we have one functional dose, like we had up at on the first row. However, because that single dose is not enough to create that wild type phenotype, we still exhibit this mutant phenotype. This is especially true in proteins that form complexes. So we're going to look at this second row here. So in the normal, again, the wild type, we see that we form this dimer. This protein forms two halves of a functional whole protein. And again, we see this wild type phenotype. In the double mutation, we still get protein product, unlike in our previous example where we had no protein. However, you see they're all misshapen and different from the wild type. And again, we see a mutant. And in our third example here, we see that with a wild type and a mutant copy of that allele, one from the mother and one from the father, we get this complex that is completely missing or is malformed and is not able to function the same way as we saw on this top row up here. And so from that, again, we have this mutant phenotype because of this mismatch of a wild type and a mutated or a recessive copy of the gene. So another layer that we're going to add to this Mendelian genetics is incomplete dominance. So incomplete dominance is this idea of taking two individuals that have kind of stark phenotypes. So in this example, we have a white flower and we have a red flower, and that breeding them can lead to an intermediate phenotype, in this case, a pink flower. And the reason this is kind of, if we look back at the incomplete dominance or the haploinsufficiency, where it's related to dosage. And so if you have two copies of the dominant allele, and say in this Punnett square, you're going to have a red flower. However, if you only have one copy, you have half as much of that red pigment, right? And so you have a pink flower. And if you have two copies of the recessive allele, as the case is down here, you have none of that red pigment being produced, and you have a white flower. So in a cross of a heterozygote, so a pink flower with a pink flower, we would have 1/4 that are red, 1/2 that are pink just like the parents, and then 1/4 that would be completely white. Another derivation from that Mendelian pattern of inheritance is codominance. And so codominance is when both alleles of a heterozygote are expressed. A good example of this is blood type. The different combinations of the three alleles from blood type lead to 4 different blood types. So you can have the A allele, you can have the B allele, or you can have just the I allele. And so if you have one copy of A and one copy of I, you are a blood type A. If you have two copies of A, you're A. Two copies of B, you're B. One copy of B, you're B. But if you have one of A and one of B, you're blood type AB. And then having no copies of those specific receptors, if you're blood type I, or the two alleles of I, you are technically blood type O. So this is an example of codominance where both version A of the allele and B of the allele are expressed together and equally shown on the protein. We can take our codominance example a little further related to blood type and look at sickle cell anemia. And so in sickle cell anemia, each allele of a heterozygote is expressed, and it's evidence with the shape of this blood. And so if you have two copies of the sickle cell gene, or HBS, this causes a fatal condition where your blood cells are all 100% of your blood cells are kind of crushed into that sickle shape and is not able to support an adult human. And so in this gel here that we see that this first lane of the gel, we have somebody that has normal blood type, so just the HbA, the normal hemoglobin. And then in the second cell, or the second lane here, we have somebody that has 2 copies of sickle cell anemia. So their cells are a lot smaller and are a lot more malshapen and that sickle cell shape, so it causes them these pointy cells don't migrate as far in the gel, and so they don't travel as far. And you see that there's only 1 blob in that lane, meaning that they have 2 of the same copy of that allele. And then lastly, we have somebody that is heterozygote, so they have 1 copy of the sickle cell and then 1 copy of the normal trait here. And so they have 2 blobs on this lane, or 2 marks where this blood has migrated on this lane. And so they're a carrier of sickle cell, and they exhibit sickle cell trait, but they have 1 functional allele as well. And this functional allele is enough for them to function normally, and they just exhibit sickle cell trait instead of actually having the anemia. So a lot of our previous examples here, we've had a single gene that has 2 alleles, but a single gene can have a whole bunch of alleles, as we talked about in a previous chapter. Depending where the mutation is or the SNP is within that gene, you can have a whole bunch of different alleles. And so in this example of clover leaves, we see that a single gene can have a whole bunch of different alleles. We have an I allele here, we have an H allele, we have an F allele, and then the different combinations of these alleles all lead to different phenotypes depending on if you have 2 copies of B, a copy of L and B, a copy of H and B, etc. And so all these different combinations of different alleles from a single gene can lead us to a whole bunch of different phenotypes. So when we've talked about our Mendelian inheritance, we've talked about our ratios of 3:1, 1:1, 9:3 to 3:1, 1:1 to 1:1, etc. But we have not yet talked about how you achieve a 2:1 ratio. So when we cross 2 yellow rats, we can actually see this 2:1 ratio in action. And so if you cross a yellow rat and another yellow rat, we end up with a Mendelian pattern of inheritance. However, we notice that here's the parents' genotypes. So we have an individual that's heterozygous that has 1 copy of the yellow allele and then 1 copy of the black allele, times an individual that has 1 copy of the A allele and 1 copy of the black allele. And we end up with this normal Punnett square where we have 25% of the offspring that are homozygous for the yellow allele, 25% that are homozygous for the black allele, and then 50% that are heterozygous for the yellow and black allele. Now, if we look at the phenotypes, we see that if you inherit 2 copies of that yellow allele, it's lethal. And so those individuals do not make it to birth. Their embryos aren't viable, and they end up getting terminated before they even start to develop. Now, if you are a heterozygote, you are a yellow individual. So 2 parts of the offspring are this yellow phenotype. And then if you are homozygous for the recessive allele, then you are black coat color. Now, we call this a recessive lethal allele. However, if we look at our actual product of this cross, you'll notice that in the heterozygote, if you have 1 copy of Y, then you are yellow. So Y has to be the dominant allele here, right? However, the ones that show the lethal phenotype are the homozygous dominant. So wouldn't we call this homozygote dominant lethal? Actually, we do not. So anytime there is an allele combination where you are homozygous for that allele or that phenomenon is called recessive lethal. So you can be a dominant allele, but if you end up being a dominant lethal, we just call you recessive lethal. It's kind of this weird quirk. So it's possible to be a dominant allele or a recessive allele. If you cause that lethal phenotype, you are recessive lethal. Now, the other thing you may ask about this example is why the heck does coat color matter? Why if you get too much color are you terminated in early development? Well, this is actually a case of something that we call pleiotropy, or a pleiotropic event, and that's when an allele has different properties or different phenotypes within an organism. So this gene that is causing these mice to be either yellow or black coat color also may play a role in the development of the heart or the development of neurons or some other biological effect. And having two copies of that mutated allele ends up being lethal to that mouse or to that rat. And so while we can visualize it with this coat color, the actual biological effect that is causing this lethality is pleiotropic, and so it has some other function that we're not sure about. So another example of this recessive lethal is taillessness in cats. This might be a little more familiar to you than rat coat color. And so having two copies of the dominant allele of this tailless gene results in lethality in cats. And that's because the mutant allele causes spinal deformations, which results in no tail. And so having one copy of that deforming gene is okay, but if you have two copies, then that embryo is no longer viable. So now we're going to move on to gene interactions and how do we test or infer gene interactions. And so we have three steps. The first step is to obtain single gene mutants and test for dominance. We can attest these for mutants for allelism. So do we see those 3-to-1 ratios, those 9-to-3 to 3-to-1 ratios that we saw from our Mendelian crosses? And then in our third step, we combine the mutants in pairs to form double mutants and see if these genes interact. So we're going to look at an example of testing for gene interaction using this, the blue flowers of the harebell plant. So we start with this blue flower where we have two different genes, W1 and gene W2. And in this case, the plus means that we have the wild type for both gene W1 and gene W2. Now, we have three mutant individuals, and we want to see if there is an interaction between these genes, if these mutations are on the same gene or if they're on different genes. We're going to denote these mutant $, mutant £, and mutant yen sign. So, in mutant $, we have two mutations in gene W1. In the £, we have two mutations in gene W1, but they're at a different location. And then in the yen sign, we are showing mutations in W2, but not in gene W1. Now we want to do what's called a complementation test. And what this is a way of determining if the mutation that is causing this flower to be white, mutant $, is that mutation in the exact same gene as the mutation that is causing this flower to be white in the case of £. So in this case, the F1 generation, if we were to cross $, to £, is we still have a white flower. And because those two individuals, the two parents, were homozygous for those mutations in gene W1, gene W1 in the offspring, even though the mutations are in two different spots, it still has two mutated genes, and thus there's no complementation. We don't rescue this original wild type phenotype, and we still have mutated offspring. So let's explain this through a biochemical pathway. And so in this example, we have this compound called colorless precursor 1. So it's just this little molecule floating around in the flower. And generally, this gene W1, the role of that gene is to turn colorless precursor 1 into this second compound. And then gene W2 will turn this second compound into this finalized blue color here, right? And so because W1 is mutated, there is no second precursor for gene W2 to turn into this blue color. So thus, we have an absence of this pigment and we have white flowers. So let's look at our second complementation test here. So again, we're going to use two of these mutants. We're going to use mutant pound sign, and we're going to cross that with mutant yen sign. And because these two genes, or these two mutations, are on two different genes, and they're still causing this white phenotype, complementation shows us that the mutation causing pound sign and the mutation causing yen sign, that they're different for those two individuals. That one must be on gene W1, as in the case with pound sign, and one must be on gene W2, which is in the case of yen sign. So we're going to explain this rescued phenotype with this biochemical pathway, the same one that we used in the previous example. So in the case of the F1 generation of the second cross that we did, we have one functional copy of gene W1 given to us from the yen individual, and that is enough, it's haplosufficient, a term that we used previously, to convert this colorless precursor 1 into colorless precursor 2. And because we have one functional copy of gene W2 from this pound sign individual, it again is haplosufficient, and that is enough to change colorless precursor 2 into this blue pigment that we see. So we know that although mutant pound and mutant yen homozygous for their own mutations, if we were to cross them together, we show that through complementation that those two mutations had to be on different genes, and thus haplosufficiency shows us that we can kind of pull apart whether the mutations are the same in two individuals when we started off not knowing that information. So we're going to talk a little bit more about these interactions and some of these ratios that we are going to see arise that are different from those Mendelian ratios. So if we have genes in the same pathway, we end up with a 9-to-7 ratio. So we don't end up having 9-to-3 to 3-to-1. That 3 portion, that 3 portion, and that 1 portion, those all get combined into the same phenotype. So in this example, we have the normal phenotype where we have two genes that are part of the same pathway. So we have gene R here that gets transcribed into mRNA and turned into a protein product. And that protein is a transcription factor that is responsible for turning gene A into mRNA and then into a functional protein of some sort. So now, if we have a mutation in this first gene, R, and we no longer have a functional protein coming from R, we still get mRNA. However, the protein produced that was a transcription factor doesn't work anymore, and so it stops. And then protein A is never made because it was requiring that protein R was functional to make protein A. Another situation we could have is if there's a mutation in A. And so R is able to be transcribed into mRNA and creates that transcription factor. And that transcription factor works just fine. It goes over to the A gene and it makes A into mRNA. However, that mRNA has a mutation in it. So we see up here, it's kind of hard to see on this, but there's a mutation right here in this A gene, and thus there's a mutation in the mRNA that's transcribed, and so we end up with a non-functional protein. And so just like this case above it here, we have now two non-functional proteins in two of our cases. Lastly, we can have a mutation in both genes. So if you look at this in a Mendelian way, this would be, we'll say big A, so this is the homozygous dominant. This would be the example of a heterozygote. This would be a heterozygote where we have the small a's being mutations, and then this is a small a where we have mutations in both genes. So in this case, we end up having a non-functional transcription factor, and even though A has a mutation in it, it never even gets made in the first place. So if we were to look at these from Mendelian ratios, we would have 9 as the normal component, 3 to 1. However, if we look at this from a phenotype output, we have 9 normal individuals, but those 3 to 1, all 3 of these individuals do not make functional proteins. The A protein is missing in all the cases. So this is a mutant phenotype, but they all, even though their function of how they got to be mutants is different, we combine them all, and so we get this 9-to-7 ratio. I kind of jumped the gun in my last slide by drawing everything out, and come to find out there's a nice slide right after it that shows you this stuff that isn't all handwritten. But again, this slide shows the combining of the 3, 3, and 1 portions into a 7 portion to get that 9-to-7 ratio. So the next deviation from Mendelian ratio we're going to talk about is recessive epistasis. And so recessive epistasis is apparent from this 9-to-3-to-4 ratio. So you can imagine that one of the 3 and then that last 1 ratio from the 9-to-3 to 3-to-1 are combined to make a 4 ratio. And so we're going to look at a flower example again here. We have this flower that is a dihybrid, and we're going to do, so 2 genes are involved, right? We have this W gene, where W plus is wild type, and an M gene where M plus is the wild type, and then just W and the M are the mutants themselves. What W does is it converts this white pigment to a magenta pigment, and then what the M allele does, the M plus, is convert magenta to blue. So if we were to do this Mendelian ratio, or this selfing of a heterozygote, an individual is heterozygote for the W and M allele, we would normally get a 9-to-3 to 3-to-1 ratio, where we would have 9 individuals that were heterozygote, so for both alleles, I should say. And so they're able to convert this white to magenta, and then magenta to blue. Now, 3 out of the 16 individuals would be heterozygous for the W allele, so they would have 1 copy of the non-mutant enzyme that would convert white to magenta, so we see that here. But it is homozygous recessive for the mutant allele, so it has no second enzyme here to convert that magenta onto blue. Now, in the case of this second individual, it's kind of the opposite of the other 3 portion, right? So it's heterozygous for the magenta, or the M allele, but it is homozygous recessive for the W allele. Now this becomes a problem because this is kind of a pathway like we saw previously. So because there is no functional W allele, there's nothing to take this white pigment and turn it magenta. And since that's missing, even though we do have a functional copy of this M plus allele, there's no possible way for that to turn into a blue pigment because we don't have magenta pigment. And this M plus enzyme only will work with magenta pigment. It needs that to be made first before it can make blue. It can't go back and do the job of that W enzyme and make white into magenta. It only works with magenta already there. So even though that M plus is there that can make blue, we still get a white flower. Now, in the last portion, we're homozygous recessive for both of those genes, right? So not only is white not being made into magenta, even if it was, there would be nothing to make that magenta into blue. So we have another white flower. So those last 2 proportions, because they're in the same pathway, they both end up with white flowers, and that's recessive epistasis. So an example of this outside of plants, I'm sure you're getting sick of your plant examples by now, is with Labrador retrievers. And so this 9-to-3-to-4 ratio is seen in labs there's a B allele which codes for black or brown. So the dominant black is black, the recessive is brown, but there's a second allele, this E allele, and that E allele is responsible for actually depositing that pigmentation into the coat. So it gets made, but it needs this E allele to come in and actually put it into the fur of the dog. And so if that dog lacks a dominant E allele, then you have the blonde retrievers or blonde labs, the yellow labs, as opposed to either the black labs or the chocolate labs. So if we have recessive epistasis, it would only make sense that we also have dominant epistasis. And so dominant epistasis is where we combine the dominant portion of that 9:3 to 3:1 ratio, that 9 portion, with one of the 3 portions, right? And so we end up with a 12:3:1 ratio. And so again, going to flowers, we see that we have a D and a W gene. The uppercase D here is the mutant. And then the lowercase d is lighter red. So we have the mutant alleles dark red, the wild type is light red. And W, the wild type W, allows the synthesis of this red allele. And the mutant confines the synthesis only to these little spots that you see here. So if we do this dihybrid cross of heterozygotes, we end up with this 9:3 to 3:1 ratio that we saw before. And so our 9, the normal dominant allele, is where we have the dominant mutant produces dark red individuals, but the W confines that only to these spots. So we have white flowers, right? The next portion down, we have a 3 portion where we have 2 copies of the lowercase d, so we would normally be light red. However, this W mutant again confines it only to those spots, and so we have a second white flower. In this 3 portion down here, we have the dark red phenotype because we have that mutant uppercase D, and since we have 2 of the wild type, it allows the depositing of this pigmentation throughout the flower. So we end up with a dark red flower like we're seeing on the far right hand side here. So we have dark red. And then in the homozygous recessive for both individuals, it only produces that light pigment, but it does allow it to be deposited. And so thus we end up with this, they say light red, to me that looks pink, but we'll go with their annotations, light red flower. So these top 2, this 9 and this 3 up here, those get combined into a 12 portion. So we end up with a 12:3:1 ratio. Next in our deviations, we have suppression. So in this example, we have 2 different genes, M and S plus, or M plus and S plus, and these are the wild type. And so when expressed in their wild type form, these 2 genes create proteins that work together to form this protein complex. Now, if there's a mutation in the M gene, specifically if it changes the conformation of the protein, or if that is in the binding site of the protein, then we end up with no longer an active protein complex, because you can see in this indication here, the binding site no longer match on both proteins. So it doesn't come into complex, and thus it's not active. However, there is a suppressing mutation that's possible. So in this case, we have a mutation to M and a mutation to the second protein, S. However, those mutations worked out where the mutation to S suppresses the mutant phenotype of M. And so it's just this happenstance where the mutation caused a change in the binding site of one protein, but also a second mutation caused that change in a second protein. and then they're still able to come into complex. So if you look here, the binding sites, one's a triangle, and then in our wild type, it was a square. So it's just random, this coincidence that the mutations both occurred and caused this protein to still work together. Lastly, we can have a mutation only in the suppressor, or in the second protein, I'm sorry, and there is no mutation in the first protein. And so this is kind of out of order the way that they did this, but so this would be our 9, this would be a 3 portion, this as well is a 3 portion because there's only single mutations, so they're heterozygote, and then this is the homozygote recessive, right, because it's got 2 of the mutated alleles. And so if we were to combine these two, because they both have active, bright, sunny complexes here that are shining bright, we end up with a ratio of 10. And if we were to combine the 2 non-functioning complexes, the 2 phenotypes that show no function, we would end up with a 6. So this ends up with a 10:6 ratio. Okay, so now we're going to talk about synthetic lethality. That is when the double mutant portion ends up exhibiting a lethal phenotype. So in this example, we have this protein complex of A and B, where both of them are binding to DNA to have via transcription machinery or whatever they end up doing. And the A portion forms these 3 hydrogen bonds, and the B portion forms 3 hydrogen bonds with DNA, and that keeps it stuck to the DNA. Now, if you have a mutation in this A protein, because A and B are in complex together, the B is able to kind of hold it to the DNA still, right? So you have this leaky function that we talked about before, this leaky phenotype. So it's not the same as this portion. This is the normal phenotype, but we have this leaky phenotype. And now if you have a mutation in B, with A being wild type, A is able to hold it to the DNA still, and you might have a little bit different phenotype. It still will work in a way, but you will still see that mutation in B. However, if you have 2 mutations, we no longer have the ability to function as a unit. It's no longer able to bind to the DNA and provide transcription, etc. And so we get a lethal phenotype. So we don't see this portion ever. So this is a 0. So this is how we end up getting a 9:3 ratio. So here is the table of just some of them. You see that the 9:3 ratio is not on here, but these tables, these ratios are going to be very important for you to know. So 9:3 3:1, there's no interaction. 9:7, genes are in the same pathway. 9:3 4, there was recessive epistasis. 12:3 1 was dominant. We didn't really talk about this suppressor has no phenotype. So I might ask you what, it might come up in a matching what a 13:3 ratio is, but we didn't go over any examples. And then ignore this is like mutant part, but the 10:6 ratio was the suppressor example. So on top of these different ratios, there's also things that lead to these diverse phenotypes you see in people. No 2 people look the same. And one of these things is penetrance. And penetrance is this idea that you can have a gene for a disease, for example. However, sometimes people carry that disease and it's not as bad, or they don't even show phenotypes or any sort of symptom, right? And sometimes they'll have a little bit of a phenotype, and sometimes they'll have a very stark and very bad phenotype. So in this pedigree here, we see that we have a person that has this disease, and then here we have a person that is a carrier, so they have the allele, but they don't show these phenotypes. And that person has kids, and then 2 out of 5 of those kids, they show penetrance and have the phenotype as well as the allele. So sometimes you can have the disease allele, but not show any phenotype, and we call that incomplete penetrance. So like that last slide, I want to distinguish between penetrance and expressivity. So penetrance, like that last slide, is you have the allele, but you don't show the phenotype, right? So you have an allele for baldness. Thank God I'm not there yet. But you have an allele for baldness, but you just never show it in your lifetime. Your dad has it, your kids have it, but you just never showed it. So that was incomplete penetrance, right? Or variable penetrance. Now, variable expressivity is the degree. So penetrance is yes or no, it's binary. You either show it or you don't. Now variable expressivity is how bad you show it, right? And so you have, if I have this bald example again, people might not say that I have variable penetrance because my dad's got very little hair, and I know that that baldness isn't a single gene thing, but use it for this example. So they would maybe say for variable expressivity, okay, you know, you're not bald, but you have a damn high hairline, so I might be showing a little bit of that phenotype, that expressivity, as opposed to being not completely bald. I definitely don't have, you know, a model's head of hair. So that might be variable expressivity. And you can have a combination of both. So you could have variable expressivity and variable penetrance. So there could be some individuals that have the allele and show no phenotype for that allele. And so those are the ones that have the variable penetrance. And then of those people that do show penetrance, there could be a range of expressivity in those individuals. So, you know, it's not as binary. There's some people that don't have it at all, but the people that do have it show it in different degrees. And so that would be a combination of penetrance and expressivity. So we'll go back to a dog example, even though my hair example I thought was pretty good. But an example the textbook gives is with dogs, and so beagles specifically. I love beagles. I wish I had a beagle. My wife was not down for the howling and yipping that they do, so we got a mutt. But anyways, back to the lecture. So beagles, they have this spot on them called piebald. So the piebalding spot is this black spot that I'm kind of trying to circle here on this first beagle. And so if you either have that spot, that gene, or you don't have that gene, but it shows variable expressivity. And so even the individuals, when they do have that spot, the amount of black that they have on them varies completely differently between individuals when they do show that the penetrance of that allele.